Very good morning, students. Let us start today's class with a prayer. God, grant me the serenity to accept the things I cannot change, courage to change the things I can, and the wisdom to know the difference. Amen. Dear children, we have seen about microorganisms in the previous class up to yeast. Now we are going to see about algae. Algae are very simple plant like eukaryotic organism. You know the meaning of the eukaryotic organism. They are found in moist habits. They are rich in chlorophyll so that they can prepare their own food. Can be seen as thin film on the surface of lakes and ponds. Therefore, they are known as grass of water. They are autotrophic and they are produce their own food with the help of chloroplast. Chloroplast contain chlorophyll for photosynthesis. The study of algae is called algology. The algae varies in size. It varies from 1 micron to 50 meters. Micron means 10 to the power minus 6 meter. Very, very, very small. You cannot see in your naked eye. You can see only through microscopes. So that is 1 micron to 50 meter. It may be microscopic, it may be macroscopic. Example for microscopic unicellular algae is Clamidomonas. An example for multicellular and macroscopic algae is sargassum. Unicellular algae only we are going to see. It may vary in shapes. It may be of spherical and rod and spindle shapes. Multicellular algae are mostly form as filaments and branches that's like thread. About the multicellular algae we are going to see later. Now in this lesson we are going to see single cellular unicellular organism, that is Clamidomonas. Clamidomonas is a very simple cell. Unicellular motile freshwater algae. What do you mean by motile? Motile means it can move from one place to another. They are oval, spherical and pyriform in shape. What do you mean by pyriform? It is in pear-like structure. And it is commonly found in ponds and ditches and water tanks. They have narrow anterior end and broad posterior end. The cell is surrounded by a thin film cell wall which is made up of cellulose. This green color outer layer. This is cell wall. It is made up of cellulose. Cellulose are found in plants which produce their own food. The cytoplasm is seen in between the cell membrane and the chloroplast. What do you mean by chloroplast? This is the chloroplast and this is the cytoplasm. Light green color shown in your figure is cytoplasm. The cytoplasm is seen between the cell membrane and the chloroplast. The cell contains large dark nucleus lying inside the cavity of the cup-shaped chloroplast. This is the cup-shaped chloroplast and this is the nucleus. This is the nucleus which is inside the cup-shaped chloroplast. The anterior part of the cell bears two flagella. These are flagellas which help in locomotion, help to move from one place to another, and two contractile vacuoles are seen at the base of each flagellum. The plural for flagella is flagellum and these are the vacuoles. The anterior side of the chloroplast contain a tiny red color eye spot. Here is the eye spot, tiny red color eye spot. Clamidomonas exhibit sexual and asexual mode of reproduction. Next we need to see about Protozoa. What do you mean by protozoa? A protozoan, in Greek, protos means first and zoon means animals. So, it is a single celled eukaryotic primitive cell, that is eukaryotic cell. They are included under the kingdom Protista. You are going to see about the kingdoms in 9th standard. So, Protista. The study of protozoa is called Protozoology. They are found in ponds, ocean, in moist soil and the cells and tissues of plants and animals causing diseases. They ranges from 2 to 200 microns. They are only microscopic. The protozoans have specialized organelles. These organelles are used for movement, feeding and other functions. The type of protozoans, there are various types of protozoans. It depends upon this cilia for locomotion. So they have seen three types of protozoan here. There are four types basically. Presence of cilia for locomotion. Example, paramecium. See the hair-like structure around the outer layer. Presence of flagella for locomotion. This is the flagella. As you seen in the case of Clamidomonas, only one flagella. This is euglena. Example, presence of pseudopodia for locomotion. Pseudo means poly, nothing. It seems to be nothing having. The cell structure itself turned like a leg for the locomotion. Other times it will not act as a leg. Example, amoeba. You are going to see about amoeba in this lesson. Next, parasite. They have shown the picture of a parasite, plasmodium, in your textbook. So we are going to see about the protozoa called amoeba as an example. Amoeba is a unicellular microorganism. It is found in ponds. Amoeba is irregular in shape. You can draw any zigzag round for drawing amoeba. Amoeba is irregular in shape. It has cell membranes, cytoplasm and nucleus. It has cell membrane, cytoplasm and nucleus. It is a protozoan that moves by means of pseudopodia. In Latin, false feet. Pseudo means false, podia means feet. So, pseudopodia or the extended part of cell membrane. It helps to catch its prey. Algae are the mostly important prey for this protozoa, amoeba. The body flows around the food particles and engulfs it, forming food vacuoles. Contracted vacuoles are seen in the cytoplasm that helps in excretion. So, excretion process is done through this contractile valves, vacuoles. Amoeba reproduces by means of fission and sporulation. Fission, it can split into two parts. Sporulation, spore-like structure created in amoeba and it fell down and again it grows as a new amoeba. That is sporulation. Next we need to see about prions. The word prion is derived from proteinoxous infectious particle. Prions have neither DNA nor RNA to transmit infection. A prion is a muttered form of a usual harmless protein. It is a harmless protein. Prions causes diseases by affecting brain or neural tissues. So another example is kuru associated with cannibalism. Next, virions. Virion is an entire virus particle, consists of an outer protein shell called capsid and an inner core of nucleic acid, RNA or DNA. If the virus is found outside the cell, it is known as virion. Outside the cell, it is dead. It is act as a dead. When it enter into a human body or other living tissues, it act very vigorously. So we cannot clarify virus as a living or non-living organism. It is different. Uses of microorganisms. Next we are going to see the topic uses of microorganisms. Microorganisms are useful in different fields such as medicine, agriculture and industry. Some of the uses are given for in your 8th standard. First, medicine. They are used in two ways. One is antibiotic, another one is vaccine. Anti means against. Antibiotic is a substance produced by living organism which is toxic for other organisms. Antibiotic, I repeat, antibiotic is a substance produced by living organism which is toxic for other organisms. So these kind of antibiotics first invented by Sir Alexander Fleming. He is the first person to discover an antibiotic called penicillin in the year 1928. 1928 he discovered penicillin. It is an antibiotic for the disease tetanus and diphtheria. Another name of antibiotic is streptomycin, which is derived from a bacteria which is used to cure various bacterial infections like plague. Next, second importance, medicinal importance is Vaccines. Vaccines are prepared from dead or weakened microbes. Edward Jenner is the first person to discover smallpox vaccine. Very important thing in the medical history, the discovery of smallpox vaccine. It is discovered by Edward Jenner. He coined the term vaccination. So, when the vaccine is injected to the body of a patient, the body produces antibodies to fight against the germs. These antibodies remain inside the body and protect from the future problems due to the germs. Therefore, vaccination is the other word called immunization. Example, in from our childhood, number of vaccines we are undergone. MMR, BCG, these are all the name of the vaccines. Next importance of microorganism is in the field of agriculture. First uses natural fertilizer. Microorganisms are called as decomposers because they act upon the degradable waste. It cannot degrade non-degradable waste. Plastic and all non-degradable waste, we cannot degrade them using microorganisms, but now we have produces tons and tons of non-degradable waste and non-degradable waste around the world. So degradable waste can be decomposed using these microorganisms. During the process, nitrates and other inorganic nutrients are released in the soil, so which in turn makes the soil fertile. This compost is called as natural fertilizers. These are the biofertilizers given in the book. Okay, rhizobium, very important. Rhizobium is a bacteria. Okay, biofertilizer. You will learn them in later classes. Nitrogen fixation. Nitrogen fixation, rhizobium bacteria living in the root of the nodules of the leguminous plants enrich the soil by fixing the atmospheric nitrogen as nitrates, which are essential for the growth of the plant. So for a plant to grow, nitrogen is a very important essential nutrient. But these nitrogens are only available in atmosphere. You know about the composition of atmosphere, 71 percentage nitrogen, 78 percentage nitrogen, 21 percentage oxygen, remaining 1 percentage carbon dioxide and other non-important gases are there. So these all nitrogen is available in atmosphere only. So to grow, the nutrients should be available in the soil. What is the work of this rhizobium? It absorbs the atmospheric nitrogen and it make the nitrogen as nitrate in the soil and helps the plants to grow with complete nutrients. Some free living bacteria in soil like cyanobacteria can also fix nitrogen biologically. Next, third importance, agricultural importance is biocontrol agents. Microbes are used to produce the crops from pest. Some of them are given below. Bacillus thuringius helps to control insects. Seagulls. Next, trichoderma fungi helps to protect roots and control plant pathogens. Bacilloviruses attack insects and other arthropod. In your figure 16.15, they have shown the picture depicts the BT, Bacillus thuringius detaili. I have a thinking thought about this Bacillus thuringius. Whichever given in your textbook, you just go through the practical usage, practical application or plus point or negative points of that content. This Bacillus thuringius is given in the advantage of the microorganisms, but it is not an practically it is not an advantage. Many disadvantages are there in the BT variety, BT brinjols, BT cotton, BT number of BT varieties plants were be available in the market, but few of them were banned due to some harmful effects. So go through the net and find out the advantage as well as the disadvantage of these BT varieties. Next, industry, sewage treatment. Aerobic microbes are allowed to grow in the primary influent during the secondary stage of wastewater treatment. Wastewater treatment is a very important thing. Here in the secondary stage, aerobic microbes are used. Those are nitrobacterium and anaerobic treatment of sewage, methanobacterium. These are all the bacteriums used in the sewage industry. And protection of biogases, human and animal waste and plant waste are broken down by anaerobic bacteria to produce methane. Methane in turn is known as biogas. Along with carbon dioxide and hydrogen, these bacteria are called methanogens. So biogases are very important. We can produce biogases using waste. It is a very important scientific process. Then production of alcohol and wine. Alcoholic drinks are prepared by fermentation process using yeast. Sugars present in the grapes are fermented by using yeast. Beer is produced by the fermentation of sugar in rice and barley. Microbes in retting and turning. Retting means to remove, to loosen the tightening of the threads. Flax plant are tied in bundles and kept in water. Bacteria loosen the supporting fibers of the stem. It loosens the supporting fibers of the stem by acting on the stem tissues. This process is known as retting. So by making it as fiber, we can use those fiber for any other purposes, like producing jute bag, jute earrings, jute sarees, jute chudidars, any jute bags, etc. Okay, linen thread is made from these fibers. Then in turning industry, bacteria act upon the skin of the animal and make it soft, and therefore it become pliable. Pliable means easy to bend or shape. So to make something brown and soft is called as turning. To turn something brown and soft is called as turning, and by this process turning we can make the skin of the animals to be bent according to our wish. We can prepare handbag, leather belt, shoes, etc. using this animal's skin. In daily life, making bread, we can prepare bread. Yeast is used in that preparation. Curd is used prepared using a bacterium called lactobacillus bacteria. Human intestine, there are some microorganisms in the human intestine which helps in the synthesizing of vitamin K and vitamin B complexes. Microbes in food process. Next we have to see about microbes in food process. Food preservation is the first and foremost important thing in this food process. On talking about food preservation, there are two methods. One is traditional technique, another one is modern technique. In both the things it is used. First, fermentation. Fermentation is the microbial conversion of starch and sugar into alcohol. As we have seen in the under the title production of alcohol and wine, the fermentation process is happening. And pickling is a method of preserving food in an edible antimicrobial liquid. So we prepare pickles using microorganisms. Vinegar, alcohol, vegetable oils are used there. And boiling liquid food items kill all the microbes, etc. Example, milk and water. And sugaring, sugar is used to preserve fruits like jam, pulp, pear, peaches, crystallization form, so that we can produce dry condition. Modern techniques. Pasteurization is the modern technique. It is used to preserve the liquid food item. This method was invented by Louis Pasteur, so that process is known as pasteurization. In 1862, milk is preserved by this method. We get oven milk, isn't it? They are preserved in this method called pasteurization. So it is heated up to 70 degrees Celsius to kill the bacteria and cooled to 10 degrees Celsius to prevent the growth of the remaining bacteria. So the milk is stored in a sterilized bottle in a cold place. Next, food protection. Probiotics or food supplements used in yogurt and other fermented milk products. Lactobacillus bacterium is used here. So decrease the risk of colon cancer, decrease the cholesterol absorption, preserve diarrheal diseases by increasing the immunity. So harmful microorganisms. Even though the microorganisms are used in many useful ways, there are a lot of harmful effects in these microorganisms. So now we are going to see the harmful effect of this microorganism. Even though microorganisms are useful, they are harmful too. And in your book they have given two tables, the harmful effect in plants and the harmful effect in animals. So you go through the diseases caused by microorganisms in animals, diseases caused by microorganisms in human. They give the name of the microorganisms, mode of transmission, symptoms, and how we can prevent from the disease or how can we treat this disease. These are all given in the table detailedly. You go through it and gain a knowledge and try to lead a healthy life. Even there are harmful effects due to microorganisms. There is a close relationship between man and microbes. There is a balance and imbalance and uses. We can use the microorganism in a useful way and we can prevent the diseases with a very careful mind. So I hope you all understand the lesson. Lead a happy life. Thank you.